Module 2, DNA Repair. In this module, students will learn to summarize the principles of DNA repair. View of DNA repair. Cells have different types of systems for detecting and repairing DNA damage according to the significance or degree of DNA damage. Some types of DNA damage may be minor, such as the net effect of a simple altered base. Other types of DNA damage are more significant, and these would include things such as double-strand breaks or DNA cross-linking, as these types of damage can block DNA replication or transcription. Different molecular sensors exist within the cell to detect DNA damage and trigger an appropriate DNA repair pathway. If the DNA lesion is substantial and the initial repair is not effective, cell cycle arrest may be triggered that temporarily shuts down the process of DNA replication and cell proliferation. In other cases, apoptotic or cell death may be triggered that would remove the cell with the damaged DNA. These types of checkpoint cell cycle and apoptotic processes are often dysregulated in cancer. The DNA repair process occasionally involves a simple reversal of the molecular steps that causes the damage. However, this is rare in human cells. Instead, in human cells, normally DNA repair pathways do not involve direct reversal of the DNA damage, but instead rely on one of several alternative DNA repair pathways. Most of the time, repair needs to be made to only one strand of DNA, but sometimes both strands need to be repaired, as in the case of interstrand cross-linking or double-stranded DNA breaks. This table shows the various types of DNA repair, the type of DNA damage or alteration that it is specific for, and describes the DNA repair mechanism by which that type of DNA damage is repaired and any additional comments. It is important for students to become familiar with this, but you can see that there are several different types of DNA damage that can occur, as well as several different types of DNA repair mechanisms. So we can consider base pair mismatches that are caused by replication errors, and these are repaired by the mismatch repair system as detailed in section 10-3. We can also have DNA damage such as small insertions and deletions due to DNA replication slippage, and these are also repaired by the mismatch repair system. With respect to the modification of bases, we can have small-scale single-base modification such as oxidation, deamination, methylation, and these are repaired using the base excision repair pathway. This would also be true for a single base deletion or an abasic site that's generated vis-a-vis hydrolysis, and this too would be repaired by the base excision repair pathway. If we have bulky helix distorting DNA lesions, such as large DNA adducts, DNA interstrand cross-linking, these are repaired using the nucleotide excision repair pathway. And then, of course, we have the single-stranded DNA breaks, and these are repaired using a variation of the base excision repair pathway. Double-stranded breaks are repaired using primarily homologous recombination mediated DNA repair, as well as non-homologous end joining. The DNA interstrand crosslinks are repaired using the HR and Fanconi anemia DNA repair pathways. Particular slide describes in detail one of the repair pathways, that is base excision repair. The base excision repair pathway is specifically aimed at lesions where a single base has been modified or excised by hydrolysis to leave an abasic site. So the base excision repair pathway is good for modified base damage, DNA damage, or for repair of an abasic site that has been generated through hydrolysis. Essentially, the goal is to replace the modified base by the correct one using a specific DNA glycosylase that will actually remove the incorrect or the modified base. For all abasic sites, the residual sugar phosphate residue is then removed by a dedicated endonuclease and phosphodiesterase. The gap is filled by a DNA polymerase that is used to insert the correct nucleotide, and then that base is joined to the parental DNA strand using a DNA ligase. Note that the same DNA repair machinery occasionally makes a more substantial long patch repair, which involves replacing more than a single base. This slide depicts this nucleotide excision repair. Nucleotide excision repair is a mechanism that allows the repair of bulky, helix-distorting DNA lesions. After the lesion is detected, the damaged site is opened out and the DNA is cleaved some distance away on either side of the lesion, which generates an oligonucleotide of about 30 nucleotides in length and contains the damaged site, which is then discarded. Resynthesis of the DNA is performed with the opposite strand as a template. The priority is to rapidly repair bulky lesions that block actively transcribed regions of DNA. A specialized subpathway, transcription-coupled repair, initiates this type of repair after detection of RNA polymerases that have stalled at the damaged site. In addition to the two types of DNA repair pathways that we have previously discussed, that is base excision repair and nucleotide excision repair, we also should consider single-strand break repair and base mismatch repair. Single-strand break repair is relatively simple, and it's also called Nick repair, and this is caused by the breakage of a single phosphodiester bond and is quite common. These are easily reversed by the enzyme DNA ligase. More complex breaks occur when oxidative attacks cause deoxyribose residues to disintegrate. A type of base excision repair that is then employed, strand breaks are rapidly detected and briefly bound by a sensor molecule that initiates repair by attracting suitable repair proteins to the site. The gap would then be filled by DNA polymerase and DNA ligase. In base mismatch repair, this mechanism serves to correct errors in DNA replication, and these errors are important in the development of cancer, and it will be discussed in greater detail in Chapter 10 of this course. Homologous recombination mediated double-strand break repair. This slide depicts HR-mediated double-strand break repair. This is a highly accurate repair mechanism that requires a homologous intact DNA strand to be available to act as a template strand. Through a series of crossing over and recombination events, the repair is highly accurate because both broken DNA strands, the undamaged sister chromatid strand, act as templates to direct the incorporation of the correct nucleotides during DNA synthesis. Alternatives to DNA repair. Undetected DNA damage tolerance, and translesion synthesis. DNA damage may sometimes go undetected. For example, cytosines can occur within the dinucleotide CG are highly mutable as a result of inefficient DNA repair. Deamination of cytosines residues normally produces uracil, which is efficiently recognized as a foreign base in DNA and eliminated by a uracil DNA glycosylase. Deamination of 5-methylcytosine, however, produces a normal DNA base, a thymine, that can sometimes go undetected as an altered base. As a result, CT substitutions are the most frequent type of single nucleotide change in our DNA. Sometimes DNA lesions may be identified but are not repaired before DNA replication damage tolerance. For example, DNA lesions that block replication may be bypassed rather than be repaired, and non-classical DNA polymerases are required to resume DNA synthesis past the damaged site, otherwise known as translesion synthesis. Subsequently, the gap in the daughter strand opposite the lesion is filled in. The lesion can be repaired later on by using the daughter strand as a template in nucleotide excision repair. Both the non-classical DNA polymerases used in translesion synthesis enable a low-fidelity DNA replication. They have a higher success in incorporating bases opposite a damaged site, but they are prone to error by occasionally inserting the wrong base. As a result, replication forks are preserved, but at the cost of mutagenesis. This slide shows why C to T mutations are the most commonly observed in human DNA. Deamination of cytosine is a very common reaction in our cells and normally produces a uracil, a base that is unusually found in DNA but is normally found in RNA. Our cells are well equipped with a special enzyme, uracil DNA glycosylase, that recognizes uracil in DNA and then removes it as part of the base excision DNA repair pathway. However, as shown on the right, in the DNA of other vertebrates, many of our cytosines are methylated at carbon atom number 5. Deamination of 5-methylcytosine produces the base thymine, a base that's normally found in DNA. Although a stable CG base pair has now been replaced by a TG base mismatch, the base mismatch may often escape detection by the base mismatch repair system, which focuses on DNA replication events. At a subsequent round of DNA replication, the thymine will form a TA base pair and effectively produces a C to T mutation. As DNA damage accumulates in all of us throughout our lives, it is inevitable as we grow older that the incidence of somatic mutations will increase, and thus we are at increased risk for developing cancer and have a declining efficiency of various cellular processes that overall contribute to the aging process. There are over 170 human genes that are known to be involved in DNA damage responses and DNA repair. And a wide variety of single gene disorders have been described that result from germline mutations in genes that work in these DNA repair pathways. As expected, increased susceptibility to cancer and accelerated aging are often found in these disorders, but a significant number have developmental abnormalities and neurological features that are very common. Although many cell types are regularly replaced, non-dividing neurons are especially vulnerable. These have high oxygen and energy needs, and they accumulate DNA damage over very long periods. In addition to various clinical features, cellular abnormalities are frequently seen with respect to chromosome and genome instability, as listed in this table. So, for example, one form of hereditary colorectal cancer is due to a variation in mismatch repair. There are several different syndromes that are associated with transcription coupled repair, and the list goes on and on. So it would be important to review this table and cover it well. On the far right, the columns C, P, N, and I refer to the type of clinical syndromes that are associated with the loss of these repair pathways due to mutation in genes in these pathways. And those in the C column are associated with the development of cancer. P are associated with the development of progeria, which is a premature aging syndrome. N is associated with neurological features, and I is associated with immunodeficiency. So you can see that in some of these, for example, interstrand cross-link repair Fanconi's anemia, you would expect to have deficits throughout all four of those clinical parameters: cancer, progeria, neurological, and immunodeficiency. Whereas some are focused in one particular area, mismatch repair is fairly only associated with colorectal cancer. This slide depicts some patient phenotypes associated with DNA repair disorders. In A is shown individual with extensive skin cancers who is suffering from xeroderma pigmentosum, and that is a disease which involves a defect in nucleotide excision repair. Those individuals have genome instability, and they have deficiencies in their repair pathways that leads to them having a little protection against UV radiation, and exposure to sunlight induces skin tumors. In B is shown an individual, B and C are shown an individual who has Werner syndrome, which is exemplific of progeria or accelerated aging. Those individuals have multiple defects in the DNA repair pathways, including the homologous recombination pathway, the telomere maintenance pathway, and the base excision repair pathways. In B is shown the individual at age 13, and to the second picture in B is shown that same individual at age 56. Disorders that have clinical features that mimic accelerated aging have those who present prematurely with gray hair, cataracts, osteoporosis, type 2 diabetes, and atherosclerosis. And these individuals generally die at or around the age of 50 as a result of cancer or atherosclerosis. And finally, in C is shown a characteristic quadrilateral and trilateral chromosome formation that is found in Fanconi's anemia cells. And it is a diagnostic of one may think that diagnosis of a DNA repair pathway disorder would be challenging, and oftentimes it is. However, cell analyses often reveal genome and chromosome instability that is characteristic of individuals that have disorders of mismatch repair. Cells from individuals with a DNA repair disorder quite often show increased frequency of spontaneous chromosome aberrations that can be characteristic of the disorder, as shown in the case of ataxia telangiectatica, Fanconi's anemia, and Bloom syndrome, which shows very high levels of sister chromatid exchange. Chromosome analysis, as shown in this figure, can also provide a simple route to laboratory-based diagnosis. Fanconi anemia, which is characterized by the variable presence of associated development abnormalities plus progressive bone marrow failure and increased risk of malignancy, can be caused by mutations in any one of at least 13 different genes that work to repair intrastrand crosslinks, making DNA-based diagnosis difficult. Chromosome-based diagnosis is more straightforward, however. Lymphocyte cultures are treated with dipoxybutane or mitomycin that induce DNA interstrand crosslinks, and chromosomes are then analyzed for the evidence of chromatid breakage, which would produce characteristically abnormal chromosome fragments, as shown in figure here. So the bottom line is that in many DNA repair pathways, diagnosis at the DNA level using a hybridization-based method or DNA sequencing is difficult because of the large number of genes that are involved in these pathways. However, when one looks at the whole genome or chromosomes at large, using cytogenetic techniques, as is shown in this slide, diagnosis is easier because the fragmentation patterns that you would obtain from an individual who has a defective repair pathway would be different than the pattern that you would see under these types of tests with a normal individual. Sometimes DNA lesions may be identified but are not repaired before DNA replication. This phenomenon is known as damage tolerance. For example, DNA lesions that block DNA replication may be bypassed rather than repaired, and then non-classical DNA polymerases are required to resume DNA synthesis past the damaged site in a process known as translesion synthesis. This table shows the DNA polymerases that are involved in translesion DNA synthesis. In translesion DNA synthesis, subsequently the gap in the daughter strand opposite the lesion is filled in, and the lesion can then be repaired later on by using the daughter strand as a template in nucleotide excision repair. The non-classical DNA polymerases, such as zeta, eta, iota, kappa, nu, and theta, exhibit a low fidelity in DNA replication as compared to the traditional DNA polymerases. Although they have a higher success rate in incorporating bases opposite a damaged site, they are more prone to error and occasionally insert a wrong base. Thus, as a result, replication forks remain active, but at the expense of inducing mutagenesis. The other non-classical DNA polymerases, lambda, mu, and terminal deoxynucleotide transferase, are involved in immunoglobulin formation, as well as double-strand break repair and base excision repair.